In this lesson, we will examine the physiology of the eye, how it works, and its defects. The eye receives electromagnetic waves within the visual spectrum from the external world and passes them to the brain for interpretation into an image. The eye is made up of 3 layers of tissue. The tough outer layer is the sclerotic coat, 5/6 of which constitutes the white of the eye, the sclera, and 1/6 in the front forms the cornea. The cornea is transparent and emits light to the interior of the eye. It acts as a fixed focusing device and is responsible for between 70 and 80% of the total focusing of the eye. It achieves this by its convex shape, which bends the incoming light rays. The surface of the cornea is kept moist, nourished and dust-free by secretions which are slightly saline, from the tear glands located under the upper eyelid. Any excess fluid drains into the nose or overflows if one is crying. The middle layer of the eye, the choroid's coat, is deeply pigmented a dark brown with melanin, which reduces the reflection of stray light within the eye. In the front portion of the eye, this layer can be seen as the iris. It too is deeply pigmented and gives the eye its colour. The iris will be brown if a great deal of melanin, which absorbs light, is present in the front of the 4 layers of the iris. In a blue eye, there is not much melanin present in the iris, and so more white light passes through to the second layer of the iris. Here, collagen, the body's most abundant protein, absorbs most colours, but reflects back blue and grey. In the iris there is an opening, the pupil, which lets the light into the eye. The size of the opening is variable and controlled by the autonomic nervous system. In dim light, or on viewing a distant object, or if danger threatens, the pupil opens wider, letting in more light, whilst in bright light, or viewing an object close to, the pupil closes down, reducing the amount of light entering the eye and also improving its image-forming ability. The mechanism for doing this is the sphincter muscles, which form the rear 2 layers of the iris. The amount of light let into the eye in this way can be adjusted by a factor of 5:1. Before we look at the 3rd layer, the retina, we need to look at the lens. This is a transparent crystalline lens located immediately behind the iris, which minutely focuses onto the retina, the light which has already been causally focused by the cornea. The lens also separates the fluid in front of the lens, the aqueous humour from the jelly-like substance behind it, the vitreous humour. The outside layer of the lens capsule, is a clear membrane-like structure that is elastic and keeps the lens under constant tension. As a result, the lens naturally tends towards a rounder shape when not under tension, which is the shape required for close object focus. Slender but very strong suspensory ligaments, known as zonules, which attach at one end to the lens capsule and at the other end to the ciliary muscle around the inside of the eye, hold the lens in place. In order to focus on objects at different distances, the lens needs to change shape. Distant objects are viewed through a flat lens with a larger pupil, while close objects are viewed through a rounder or accommodated lens and a smaller pupil. In its relaxed, roundish, more convex shape, the lens focuses on near objects. The ciliary muscles are tense and not pushing the zonules, which are therefore relaxed and not pulling on the lens. To view a distant object, the ciliary muscle relaxes, increases in diameter and pushes against the zonules, which consequently stretch and pull on the lens, making it flatter. This process of adjusting the lens shape is called accommodation, and the pupil changes size automatically. The change is measured in diopters, and is the reciprocal of the focal length measured in meters. For example, a 20-year-old person has an accommodation of 10 diopters, the reciprocal of which is 1/10 of a metre, which means he can focus down to 1/10 of a metre, or 3.9 inches. As accommodation depends upon the elasticity of the lens, it is no surprise that accommodation becomes poorer with age, as the lens becomes harder and less flexible. This table shows how the near point of accommodation, that is, the nearest point of focus, moves further away with age, as the amplitude of lens accommodation reduces dramatically. The image leaves the lens and it is inverted and reversed onto the retina. However, the brain perceives the object in the upright position because it considers the inverted image as normal. Finally, the light, suitably focused, arrives at the rear of the eye, onto the third layer called the retina, which reflects light back onto photosensitive cells called rods and cones, which actually face away from the light. The rods and cones convert light energy from the object viewed into signals that are carried to the brain by the nerve fibres of the optic nerve. Coated onto the rods is a non-colour sensitive protein called rhodopsin, whereas coated onto the cones are proteins called opsins, which respond to different colours depending upon their amino acid sequence. Both of these proteins are related to retinol, a derivative of vitamin A, retinol, which explains why night blindness is one sign of a vitamin A deficiency. Carrots, rich in vitamin A, really are good for your eyesight. There are about 120 million rods in the retina. They are extremely sensitive to light, unlike the cones. They are able to respond to the smallest measurement of light, 1 photon, and so they are responsible for dark-adapted or scotopic vision. Although rod vision is acute, it provides only coarse images, because up to 100 rods may share a single circuit to one ganglion cell. Consequently, the brain cannot identify where exactly on the retina the light was seen by an individual rod. The rods are also responsible for our peripheral vision and the detection of movement, not only because of their immense sensitivity, but also because of their wide distribution over the retina. The eye continuously moves in order to place the object viewed in the centre of its field of vision, also known as the axis of vision. By doing so, the light from the image falls on the most sensitive part of the retina, the fovea centralis, which lies in the centre of a depression called the macula. The fovea has a diameter of only 0.3 of a millimetre, and it is densely packed with cones. There are no rods present. The cones are individually linked to nerve fibres, and so the message to the brain is an exact image of the object being looked at, and in colour. There are 3 types of cones, those sensitive to the red part, the green part, and the blue part of the spectrum, depending upon their type of protein surface. The red cones constitute about 64% of the total, green about 32%, and blue only 2%. However, the blue cones, which detect light in the lower spectrum, appear to be more responsive, and therefore compensate for their low numbers. They are located away from the fovea. From this diagram of the relative distribution of cones and rods on the retina, we can see why the rods provide the peripheral vision, while the cones provide the detailed focus or central vision. In total, there are about 6 million cones in the retina. Cones require far more light than rods in order to operate effectively, but they adapt in about 10 seconds to sudden increases in light, for example when going outdoors on a sunny day. This daylight vision is known as photopic vision. Intermediate light conditions are called mesopic. Dark conditions, where only the rods are functional, are known as scotopic conditions. Adaptation to darkness, however, takes far longer, about 7 minutes for cones and 30 minutes for rods. After a long time in the dark, rods and cones can become supersensitive to light, and even the smallest light can excite the receptors. However, this sensitivity can be completely lost by the intrusion of a bright light. Click and hold the mouse anywhere to reveal the clock on the wall, which is readable in the dark, despite its low light output.